This is the last slide set. Hmm? Oh, maybe I need to do it? Do I need to find my slides? I don't know where they are in here. No, those are not mine. It's the next one.
You had it up there for a second.
All the file names are in Hebrew. Oh, there they are. Okay, very good. Thank you very much. And thanks to Tally and to Little Steps for giving us the opportunity to come talk to you about the BRAVE study. I'm very excited to be in Israel for the first time, and this is a great opportunity for us to give a little bit of information about our trial. We named it the BRAVE study because of our belief that the boys that are suffering from DMD are very brave boys and young men. And so we're happy and eager to be joining the fight to try to produce some better medications for the DMD patients. So I'm going to talk very briefly about Mallinckrodt ARD since time is limited. Then I'm going to go through our program, talk a little bit about our drug, MNK-1411, a little bit about the study design that we're using and the scope of the study, a little bit on the objectives, a little bit of summary on some of the details of the study and how we're trying to meet the objectives, a brief coverage of key inclusion-exclusion criteria for the patients, a little talk about the endpoints, and then a current status update, and then we will be finished. Oops. So how do I make this back up?
Left arrow.
This one? Oh, this one. Very good. I didn't see that one. So just a couple words about Mallinckrodt. Mallinckrodt's actually a really old company. It's 150 years old. Probably many have never heard of it, and we actually have an inside joke inside the company, which is we're the oldest company that nobody's ever heard of. One of the reasons you haven't heard of us is we've only turned to doing autoimmune and rare diseases over about the last 5 years. So this is relatively new to the company. And our mission is to manage complexity and improve patients' lives. So on to MNK-1411. MNK-1411 is a man-made synthetic shortened version of the natural hormone corticotropin, also called ACTH, or adrenocorticotrophic hormone. It's produced in the pituitary, as you see in the diagram on the right, and it travels around the body. One of the first actions that was discovered with this compound was that the ACTH goes to the adrenal cortex, and through a melanocortin receptor number 2, it initiates production of cortisol, which is the body's own natural steroid. And so it's known to have, and it's been known for a very long time, that this drug has anti-inflammatory properties. It's used in a lot of different indications where inflammation is an issue. When the receptor was discovered to cause the cortisol release, there were also 4 other melanocortin receptors identified, a family of receptors, and they're located in widespread locations all over the body. And laboratory studies suggest that these melanocortin receptors are expressed on certain cells in the immune system, in muscle cells, nerve cells, and lots of other cell types. So we went into this project with the idea that the cortisol will produce some of the same benefits that steroids produce. So our hypothesis of how MNK-1411 may work in Duchenne muscular dystrophy goes as follows. Am I not close enough to the microphone? First of all, MNK-1411 has been tried in a phase 1 clinical trial, and it was well tolerated, but it has not yet been tried in boys with Duchenne muscular dystrophy. So this is the first study in boys with Duchenne muscular dystrophy. We know that the muscles in patients with Duchenne's show foci of inflammation around injured muscle, and that reduction of inflammation is one of the ways with with which people are approaching treating Duchenne muscular dystrophy. We also know that treatment with the exogenous corticosteroids like prednisone or deflazacort, the standard of care drugs, has been shown to slow Duchenne disease progression. So we think that MNK-1411 has 3 ways that it could possibly attenuate muscle damage and reduce muscle inflammation in boys with Duchenne. The first is direct effects on the cells in the immune system to reduce muscle inflammation. In addition to the cortisol effect that I mentioned previously, there are also melanocortin receptors on T cells, B cell lymphocytes, macrophages, and lots of immune system components. So we believe that direct effects on the immune system could be one way that we could help Duchenne. Secondary to that, as I mentioned, we do know, we're fairly confident that we're going to reduce muscle inflammation with the cortisol that's produced in this drug. And actually, I'll talk a little bit more about it later, but we've tried to model the dosing such that the high dose in this study mirrors the steroid equivalent effect of the standard of care corticosteroids. And lastly, there are some preclinical studies that show effects of ACTH and other melanocortin agonists on muscle cells in damaged muscle, and there's some reason to believe that that MNK-1411 might help there also. In addition to that, we have a preliminary experiment in MDX mice. It was published in the, a poster was presented, it was published as an abstract, and a poster was presented at the American Academy of Neurology meeting a couple of months ago, and it shows that MNK-141 reduced inflammation as measured by MRI and also in muscle histology, and that it may reduce abnormal gait that's seen in the MDX mice. So the scope of the study has grown substantially. When I first conceived of this study, it was going to be a smaller study in the USA only. But as time has gone by, we recognized that we needed a bolder, bigger study in order to demonstrate what we would like to demonstrate. And so we're proceeding with a larger trial than I originally conceived, and we are spread out across the world. This trial is a multi-center, randomized, parallel group, double-blind, placebo-controlled study, so a fairly standard design in boys 4 to 8 years of age with Duchenne's. We're going into 15 countries currently across Europe, North America, Latin America, and the Middle East, and we're going to have approximately 50 sites participating. BRAVE study objectives. In terms of objectives, the primary objective in this study is to show that boys improve on the 10-meter walk-run compared to placebo over the course of this study. Secondary to that, we're looking at the North Star Ambulatory Assessment, the other time function tests, including the rise from the floor test and the 4-stair climb, as well as muscle strength. We don't have a lot of muscle strength testing, just a little bit in this study since it's our initial proof of concept study. It's just a secondary objective. And of course, we always look at safety and tolerability. In addition to that, we have some exploratory endpoints. As an exploratory, we're also looking at pulmonary function and some surrogate markers in blood associated with DMD. Because there's likely to be an overlap, I think it's more than likely actually, between the exogenous corticosteroid effects that you see with the standard of care and the hypothesized mechanism of MNK-1411 in DMD, it's not really feasible for us to study as an adjunctive medication to the standard of care, and that's why we have a placebo group. So in terms of just giving a few more details on the study so it can be better understood, we're trying to recruit 132 boys. So this is a big worldwide study, recruiting a lot of boys. We have 4 dose arms, 2 active doses, high dose and low dose, and 2 volume-matched placebo groups. It's volume-matched because this is a big peptide molecule, so it has to be injected, and this drug is given through subcutaneous injection. Dosing is flat but weight-based, meaning that in the high-dose group, there's sort of 2 subgroups, and boys that weigh more than 20 kilograms will be in one subgroup, boys less than 20 kilograms in another, and the same for the low dose, the same for the placebo doses to keep it blinded. As I mentioned previously, the high dose has been modeled through pharmacokinetic modeling to result in cortisol exposure that's roughly equivalent to the usual dose of prednisone, which is 0.75 mg per kg. And the low dose is 50% lower than the high dose. Now, it's likely from a pharmacodynamic perspective that the low dose will produce more than 50% of the steroid equivalent effect that the high dose produces, but we're uncertain exactly how much more than. But it'll be somewhere in between 50% and where the high dose is. So patients in this study are randomized 2 to 1. So patients have a 2/3 chance of getting MNK-1411 versus the placebo in the initial 24-week study. I'm not going to belabor all the visits, but essentially there's a screening visit over a 1-month period. It's followed by a baseline visit where we see where the boys are at the beginning of the study in terms of their performance. Then there's monthly visits for the first 4 months, and then a bi-monthly visit at the end at 24 weeks. Boys that complete the 24-week double-blind period are eligible to go in an open-label extension. In the open-label extension, everyone gets drug, and they'll retain their dose assignments from the double-blind phase, meaning that if you're in the high-dose placebo group, you're going to continue on high-dose MNK-1411 in the open-label extension, and the same for the low dose. The visits are a little more sparse in the open-label extension. We wanted to try to make this a little bit easier for families and kids. So we've tried to make the dosing, the visits as sparse as we thought we could make it and still meet our objectives. So there's a baseline visit, then at 1 month, 3 months, and 6 months during the open-label treatment period. At the end of week 48, after the 24 weeks of double-blind therapy and the 24 weeks of the open-label extension, that patients exit the study, and there's a follow-up visit 4 weeks after that at week 52. So this is just for those that like diagrams, this is a diagrammatic example of everything that I just explained. And you can see here the dose groups, the placebo groups. Although, as I said, it's kind of tricky because of there's 44 patients in the high-dose group, 44 patients in the low-dose group, and 44 patients in placebo. But the placebo patients are broken up for purposes of keeping the blind into low-dose and high-dose. I just wanted to quickly go over the inclusion-exclusion criteria, what I think are the keys for understanding. Patients are allowed in the trial are age 4 to 8, but we know because we're looking for steroid-naive boys that almost all our boys are probably going to be 4 or 5 or maybe 6 at the oldest. We don't really think we're going to see any 7 or 8-year-olds. Nevertheless, we wanted to be inclusive, and if there was some patient whose caregivers had elected not to put him on steroids for some period of time and they changed their mind, that we would be open to enrolling an older patient. But frankly, I don't think we're going to get too many of those. We have a rather standard diagnosis for what constitutes Duchenne muscular dystrophy. Just to summarize, it's basically patients that have had a muscle biopsy or genetic testing that's consistent with DMD and have a clinical picture that's consistent with DMD. We are allowing approved treatments for DMD that target dystrophin gene mutations, so patients taking Adalurin or patients taking eteplirsen would be okay in our study as long as it's an approved drug in the country where you are. And also that they remain on a stable dose. In terms of exclusion criteria, patients with symptomatic cardiomyopathy are excluded, although we don't think we're going to see many of these at all in the age range that we're going to be in, which is the 4, 5, 6-year-olds. Also, patients that can't complete the 10-meter walk-run would be excluded because that's our primary endpoint. We need a baseline. Patients who had previous treatment with corticosteroids, as I mentioned, basically are excluded. However, if it was short-duration corticosteroids and the patients dropped for tolerability reasons or some other reason, we would entertain possibly enrolling some boys that were transiently exposed to steroids. We're also excluding patients with type 1 and type 2 diabetes because one thing that ACTH molecules do is increase blood glucose, and we wouldn't want to be doing that in a diabetic patient. We're also excluding patients with chronic active hepatitis or history of TB, tuberculosis, because these kinds of infections can potentially be reactivated by the immune suppression that goes on with ACTH. And lastly, for the same types of reasons, we're excluding patients that are immunocompromised. In terms of the endpoints that we're studying, this gives the whole full schedule, but basically, our primary endpoint is the 10-meter walk-run at baseline versus week 24, which is at the end of the double-blind period. And then for the North Star and the time to stand test, the time to climb 4 stairs, and the rest of the 10-meter walk-run administrations. Those are at all the other visits. We have quantitative muscle testing also at all the other visits, although it's not too laborious. We're just looking at knee flexion. And then safety and tolerability, of course. And as an exploratory, as I mentioned, we're looking at pulmonary function from time to time. So in terms of our current status of the study, as I mentioned before, we have 50 sites total that we're hoping to recruit for this study. 4 of the sites are now initiated in the US, and we're waiting for our first patient to go in. It's an exciting watch and wait. We are going to be getting rolling outside the US regulatory and ethics committee approvals over the next 6 to 8 months between this month and December. And we're currently recruiting sites in US, Canada, Mexico, UK, Spain, Belgium, Italy, Portugal, Romania, Bulgaria, Serbia, Croatia, Poland, Israel, and Turkey. I'm relatively confident that we're going to have sites. It's in the process, but I'm really relatively confident we're going to have sites in all these countries. And currently, I looked at the schedule to see when Israel would start, and it looks like Israeli sites will start probably in October, either October or November. Well, everything should be completed, and all the obstacles should be cleared to start the study in Israel. Okay. Thanks for your attention.